sorry. It's for the camera.
I will start by describing the parameters of the problem. Next, I will introduce proposed metrics from information theory that address these challenges. And finally, I'll tell you about DandD, a parameter-free tool that implements one of these metrics. The question at hand is how we measure new sequence. One way that you might go about doing this is to align your assembly to known reference in order to identify the differences. As a quick overview of that process, you start with your reads, which you assemble into larger contigs, continuous sequences formed by assembling overlapping reads. And during that process, some of those contigs may be filtered out based on their length or their amount of repetition. Next, the remaining contigs are aligned to known references, at which point the sequences that deviate from that reference by a certain percentage will be identified as new. So, over the course of this process, there are several points at which independent decisions must be made that may affect what sequence makes it to the end, to be identified as novel. When Sherman et al. Published a review of reported novel sequence findings across an array of human population studies, they found an extremely wide range of per individual sequence discovery. Not all of this difference can be explained by the diversity of the target populations. Each study team made their own decisions and threshold selections along the alignment pipeline, and as a result, it's difficult to interpret and compare the amount of novel sequence reported. So, in the interest of having
I don't know.
In the interest of having a standard metric for reporting a new sequence, it's clear that we should try to reduce the number of parameters that affect the measurement. If alignment-based methods involve too many parameters, the obvious next step would be for us to examine the processes used in alignment-free techniques. One important parameter that is ubiquitous in alignment-free analyses is K, the length of the substring that is used to as a unit in dividing the sequence into k-mers. However, the requirement that we select a K means that we have not yet fully escaped upstream decisions that affect our results downstream. As NASCO et al. Showed, the specific K that was previously properly selected for a classification task ultimately leads to less specific classification as the database grows. That's there on the left. All pairs Jacquard, which is used for sequence clustering, also depends on selecting an appropriate K, which may or may not be optimal for all parts of the task. In this example, the K that produces an accurate clustering and phylogenetic tree for one set of sequences on the left may produce an incorrect clustering for others. So ideally, we would like to remove the dependence on a pre-selected K in order, with other parameters in order to improve stability and interpretability. Why are we so concerned with identifying the optimal value of K? In this example, we use multiple values of K to demonstrate what can go wrong when an unhelpful K is used. When K is low, there's too much sequence overlap between the genomes, leading to an extremely interrelated tree. When K is too high, very few species are identified as related at all. And when we choose K in the middle of these two, we see a more classically recognizable hierarchy. On the other hand, it doesn't actually do us any good if this is not actually an accurate representation of reality. So, fortuitously, when we compare these trees to the true on-the-ground phylogenetic tree for this data set, the normalized Robinson folds distances for the low and the high K are higher than for our Goldilocks K at 10. Visually, you can see that K of 10 gives us a clustering much closer to the ground tree. All right, now we will discuss some concepts from information theory that inform our solution. In addressing the question of how much new sequence can be found in an assembly, we're aided by work in information theory related to compression, which is fundamentally related to repetitiveness and conversely to distinctiveness. One metric derived from information theory that's sometimes used to address our question is about how much new sequence there is R. R is the number of runs in a Burroughs-Wheeler transform of the sequence. If you've ever used BZIP, you've made use of some form of R. Another metric which has been used to describe difference and repetition between sequences is Z. Z is the number of phrases in the levels of parse. You have almost certainly used Z if you've ever used GZIP. I mean, I used it today. There's another metric, delta, that can be used to address this question. Delta is concerned with the cardinality of a set of k-mers within a sequence, that's S sub k. As you can see in the definition, delta maximizes the ratio between the number of k-mers and the value of k, which means that magically, k is a function of the data and not a parameter that we choose. So it makes sense that the cardinality of a set will depend on the size of k. Intuitively, the cardinality of a set in our genomics universe at k equals 1 will always be 4, because that is all of the nucleotides that are available in our alphabet. At the other end of the spectrum, imagine we had a k-mer of the same length as our entire string. There would only be one possible k-mer of that length in the sequence. So the very last point will be a 1 for some ridiculous, enormous value of k. And so what happens after that? What if k were equal to 2? Given the size and complexity of genomics data, we can make a pretty strong tradition that our sequence might contain all possible length 2 combinations of our alphabet. Depending on the data, this may also be true for several k's, and k of 3, obviously exponentially bigger, but in the world of genomics, not that long. These kind of trends give the early k's a certain shape in the graph. So it gives S of k an exponential growth for these early values of k. Because these values are equal for low k's, no matter the data set, the ratio will not be particularly descriptive. So what about the other extreme? As we said before, there can only ever be one k-mer, the same length of the sequence, which we're going to call n. In this example, our string is 6 letters long. When k is 6, we see that there's just one k-mer, one 6-mer, just as we would expect. And then working backwards, when k is n-1, in this case 5, we can see that there are only 2 5-mers possible at that length, and I've drawn boxes around them. Progressing to n-2 and n-3, we can see that logically for lengths close to n, the relationship between the cardinality and k will have a linear upper bound. On the other hand, these enormous k-mers may be of limited value when it comes to measuring similarity, given how specific the sequences are likely to be. Okay, so what happens in the middle? We find our maximum sequence cardinality and its associated k value right there in the middle, and that'll be dependent on the data, but generally there's a plateau where cardinality may be fairly similar between k's. This is where the ratio in the delta equation becomes relevant. Delta captures the point of diminishing returns before the start of the decline. Okay. Once k grows beyond the low values of k, there begins to be differentiation between sequences depending on what k-mers are present. So like a fingerprint, if there's no space between worlds and the print is just a smudge. But if there's too much space between worlds, there isn't enough information to match the print. But if there's the right amount of presence and absence, the print is distinctive. When we calculate delta, we're looking for a maximum value of the cardinality offset by the length of k. This value is greatest at the R max k. You can see a clear peak in the graph of these possible delta values drawn from E. coli genome. This is delta at R max k. It turns out that the value of R max k is based on the organism. Here's Salmonella with a slightly higher Argmax K. And intuitively, it makes sense that a more complex organism with a longer genome, such as a human, might need a longer K-mer length in order to capture the difference. So delta and Argmax K depended on the data. So recall a couple minutes ago that I introduced R and Z and delta, three different compression-related metrics from information theory, which are all based on fairly different concepts. When they are scaled, however, they mirror each other fairly closely. This would seem to indicate that they are all getting at the same underlying property of the data. The general shape of the graph is fairly linear to start before the reduction in slope, as there's less and less unseen variation with each additional genome that's added to the set. Looking over this graph, we can see that certain genomes are contributing more to the increase of delta and the other metrics, leading to a sharp jump in the measure. In our delta explorations, we call this change the delta-delta. Note that the specific order specific order of the genomes that are added to the set will influence their relative effect on the metrics. If an exemplar of a particular strain has already been added, the addition of another exemplar containing a similar sequence will have a more muted effect on the metric. When we run a real-world comparison of the wall clock time required to calculate each of these metrics for an increasing collection of Salmonella genomes, delta uniformly outperforms R, even when exact K-mer counting is used to measure delta. One of the reasons that we think delta is preferable to other measures is that it's primarily concerned with cardinality, as we discussed, which means that we can make use of a number of existing tools to quickly and accurately estimate it. DandD uses dashing by Daniel Baker. For precise counting rather than estimation, we use KMC3. Given that we're interested in measuring sequence growth, we are interested in performance that's associated with combining haplotypes. So during our analysis, we often need to union sets of previously examined genomes. In our comparisons, we pay particular attention to the average cost, like first row of each section, the average cost of determining the cardinality of a single haplotype, the cost of adding 10 haplotypes to a set one by one, and the cost of performing one single union on 10 haplotypes at once. So the benchmarking we've done shows that we can sketch each human assembly in our set in half the time it takes to count the K-mers directly with KMC3. Where we really speed ahead, though, is during this union process. Once the sketches have been made, the process of unioning all 13 or 10 of them takes moments when KMC3 takes drastically longer to count the K-mers for the unions of databases, and it doesn't really get an advantage by the fact that we've done it before. Notably, dashing's peak memory for global unions is equal regardless of the organism. The memory footprints also favor dashing in both tasks by orders of magnitude. So, now that we've discussed the issue we're trying to solve, how to measure a new sequence, and we've examined this metric, delta, which we have borrowed from information theory, it's time to describe our tools and show some of the results. Okay. So, DandD. DandD is a tool to utilize delta to estimate the amount of non-redundant sequence, content, and identify outlier contributions via their delta-delta. So, it's parameter-free, it is independent on alignment, and it's fast with low memory cost. DandD currently encompasses two different modes: a simple mode that estimates delta for the individual assemblies in a collection, as well as their union. And it can be used to answer a number of questions related to delta-delta, such as the amount of unique sequence an assembly contributes to a set. The second mode progressively unions different orderings of a collection in order to identify the function that describes the speed of collection growth. It can also identify assemblies whose contributions consistently deviate from the norm within the set. DandD is special because it can use the variations of different orderings to analyze the behavior of specific genomes relative to others. Many other tools focus on finding the mean to generally describe the shape of the curve. Orderings, so I mentioned this idea of orderings a few times. Orderings are permutations of the genomes in the set. As an illustration, when we calculate delta for sets containing single assemblies, the value for the set will be equal to that of the specific assembly that was nominally added to the set at step 1. So there's just one assembly there, and the set only contains one, and thus the deltas are different because the two assemblies are not the same. They start at step 1 with two different delta values. On the other hand, the final full union, regardless of ordering, will always have the exact same value of delta, since every ordering will be composed of identical sets in that last step. However, along the way, each ordering adds a new assembly to its set, we calculate an updated delta for each new union set. The resulting value will depend on which sequences are in that union at each step. So, for example, in these two orderings, the change in delta, the delta-delta, between sets of size 2 and 3 is notably different. The low slope for the orange line between these steps indicates that the third assembly in that ordering contributes less unseen sequence than the third assembly of the blue ordering. And it looks like my PowerPoint conversion did something strange with that last point. So just imagine that point is up there. Okay. So, alternate orderings of the same set of genomes emphasize different contributions of individual genomes. So, all right, here's a graph of values for delta of multiple orderings of the same set of assemblies. Even though they have varying shape, they still exhibit a clear trend, so it's helpful to look at the average across those orderings. And lo and behold, that's that dotted line back there. The plot is shows the sublinear function that measures how much new sequence is gained with each assembly that's added. And this is an interpretable and parameter-free way, so it would be comparable across studies. As I said previously, we can see that the order in which assemblies are added to the collection affects the contribution that each makes to the set. Here you see the exact same set of genomes added one by one in 5 different orderings. And those bumps that you see are a consequence of genomes with higher or lower amount of new sequence being added earlier than other genomes with overlapping content. It's also possible to examine different stratification of the dataset. In HGSVC dataset, we are able to stratify by variant class. First, we can see the mean curve across orderings for single nucleotide variants, and then we add indels, and finally we can add structural variants. Examining the stratification allows us to disentangle the contribution of different variant classes. Not surprisingly, the addition of indels and SVs adds a substantial amount of new sequence relative to SNVs only, and indels add more novel sequence relative to structural variants. Because they're more numerous. The previous graphs I've shown use the HGSVC data drawn from VCF files, but DandD doesn't actually require that kind of data. Previous methods of detecting novel sequence have relied on a reference coordinate system, but Delta only uses sequence, so it's equally applicable to assemblies that are just a raw set of contigs without common scaffolding. Delta doesn't need anything other than those contigs to do its work, which means that the metric automatically incorporates information regarding order and orientation gained directly from the data. On the left, left, you may recognize the mean deltas across variant types that we were just looking at in the HD SVC. On the right, we see a plot of HPRC data, the human pan-genome deltas, with 5 different orderings displayed. Why do I have these next to each other? I want to talk to you about the concept of openness, which describes sort of it characterizes the growth of a curve and sort of indicates to us whether we are open and going to find more sequence in as we go along, or whether we're closed and we're starting to run out of new stuff to see. And this is done by using Heaps' law to fit a plateau parameter known as alpha. So roughly, when alpha is less than or equal to 1, it means that the genome is open with substantial growth that's possible in the future. And when alpha is greater than 1, it means it's closed, and we've seen what we think we're going to see. So alpha fits indicate that the human brain, both human pan-genomes are open, which, we don't actually think we've seen everything, right? So that aligns with what we expect. So I promised you that DandD could be used for alignment-free methods, so here we are. On the top, you can see the traditional all pairs Jacquard approach, where, as the equation shows, the same selection of K is locked in for both steps A and B, which may not be optimal. We propose using something we're calling the K-independent Jacquard, which uses deltas in place of these K-specific values. Because the optimal values for each set are baked in, we don't need to worry about losing specificity due to our selection of K. Our next experiment relies on comparing trees produced by hierarchical clustering against a ground truth. So using the AF project databases, we compare the phylogenetic trees produced by our 2 clustering approaches to determine which is more accurate via the Robinson-Folds distance. First of all, as we promised, the Kamer count-based delta and the dashing estimate-based versions produce results that are nearly indistinguishable. Second of all, we can see that the K-independent Jacquard manages to choose an appropriate K within a narrow range of optimal Ks. So we want to be minimizing this distance, and so those lower ones are all along that same distance. Recall those 3 sequence clustering trees we showed earlier. Our Goldilocks K selection coincides with the Argmax K chosen by the K-independent Jacquard. So we've seen these exact 3 examples before, and this is how I found how I chose those K values. Note that they are different depending on our K-independent Jacquard values, depending on which K we picked, we get a better or worse tree. Now that you understand how delta works and how DandD uses delta, it's time to show you some additional use cases, and specifically the ability to address questions that are outside the scope of other methods. When we first started selecting haplotypes for our delta experiments, we took care to keep only unrelated female samples and kept the population diversity that was built into the HV-SVC2 database. But what if we hadn't done that? Here we see a collection of 7 haplotypes for largely Han-Chinese individuals, 2 of whom are related, a mother and daughter pair. We can immediately see much more variance from the mean in a number of orderings. So when these lines that are well below the mean, when an ordering falls below the mean, that indicates that the haplotype added at that step contributed less new sequence than the others did, on average, at that same step in other orderings. Put another way, we had already seen more of this individual's genome than one would expect during this ordering. Whoever got added at that step, we'd already seen a lot of what they had to offer. Sometimes that variability is really revealing. So this plot on the right across, is really showing us something very different. As you would expect, there's a clear difference in the value of delta at each step, depending on how many related haplotypes have already been included. These lines below are colored by how many related people, i.e., or how many related haplotypes, the two haplotypes of the mother and the maternal haplotype of the daughter, how many of them had been added at step 5. And the only lines that are graphed here are lines that were greater than 2.5 times the standard deviation away at some point along the process. So this is just like a subset of the lines that are outside of that. Okay, so this is actually interesting because it's actually describing a multi-way relationship between inputs that wouldn't actually be possible to be captured using all pairs Jacquard. And so, most of the methods that we've sort of mentioned so far, they're really looking at the mean, and they're ignoring a lot of information that you can actually gain by looking at the variance. And so that's what DandD has to offer. All right, so I hope I've managed to convince you that you should also enjoy DandD, since it's such a useful, fast, and parameter-free tool. In future, we plan to take DandD beyond pairwise sequence comparisons and explore using it for target discovery. And we're also interested in improving the theories that link delta to MASH systems. All right, so thank you for listening. Thank you to my lab at Johns Hopkins, my collaborators, including Omar, Ahmed, and especially to my PI, Langmead, who has given me the opportunity to do interesting work and to meet interesting people and to go amazing places. And thank you to my other lab mates who helped refine this talk. Without their support, this presentation would have just been an amusing series of TTRPG jokes and puns and